Hello children, let's move on to the next topic, factors affecting photosynthesis. So students, in the last class you have already learned about the photosynthesis and the various parts of the leaves which are responsible in photosynthesis process, which do take part in this process. So, in this module we are going to study about the factors that affect the photosynthesis. Okay, that means photosynthesis is influenced by certain factors like temperature, which is managed or is controlled by the light exposure. Then chlorophyll, carbon dioxide and the availability of the water. Students, do you know one thing? That we actually study about the rate of photosynthesis. That means how fast or how frequent the process of photosynthesis is occurring in a plant body during daytime. Okay. So in your book, actually 4 experiments has been demonstrated. So we are going to start with the first 2 today. The first one is stream of bubbles, that to see or to observe the stream of bubbles. Okay. Where do we have to observe? We have to observe these stream of bubbles in a setup given in the picture you may see. So basically we are going to study that how oxygen is formed during the photosynthesis. Okay. As in the reaction of photosynthesis, you will see that the end product is glucose and oxygen. So that means during the process of photosynthesis, oxygen must evolve from the plant body, isn't it? So to check that, certain experiments has been performed. One of the experiment or the demonstration which is done frequently in the laboratories is this, that we are going to study right now. Okay, to check oxygen is formed during photosynthesis. So for this we need a funnel, a test tube. Okay, let me mark it out. This is the test tube, the funnel, the beaker. Okay, and what we have put into it a hydrilla plant. See, we have selected hydrilla. It's an aquatic plant. We have selected this because it is used to with the aquatic habitat, and the aquatic plant do perform the process of photosynthesis in the water, and to observe the liberation of any gas in the fluid medium is easier. Okay, so that is why we have selected an aquatic plant that can be set up into the water, which is a fluid medium, and in order to perform the photosynthesis, this plant will liberate the gas, and we can very easily identify the liberation of the gas. Okay, so therefore with this hydrilla plant, what we have done, we have cut down its stem obliquely. If this is the stem of a hydrilla plant, we have cut it obliquely like this. After cutting, the stem becomes like this. So this is basically the oblique cutting. In this, why do we cut obliquely? Because it when it is cut obliquely, the surface area which is present over this part is more. We get more surface area. Okay, and the more the surface area will be, so it would be easier to observe, or it will be more, there will be more liberation of gas. Now, this water is not a plain water. Okay, now why it's not a plain water? Because we have used sodium bicarbonate. What do we have used? We have used sodium bicarbonate. Okay, so sodium bicarbonate, when it dissolve in water, it liberates out CO2, and CO2 is one of the important ingredient for the plant to perform the photosynthesis. You know that. Now, we have arranged this whole setup in such a way that the hydrilla plant is covered with the funnel at the bottom. Okay, and the cut stems are towards the stalk of the funnel, this neck of the funnel, and this neck of the funnel is again covered with this test tube, and everything is dipped inside this beaker. You may see the level of the water in the beaker, and it is dipped, the whole thing is dipped into it. Okay, so the thing, when you have kept the funnel inverted, covered the hydrilla, and when you have kept the test tube inverted on the funnel, that you have to make sure one thing, that the water should fill inside the test tube. You have to set up the whole thing in such a way. Now we will put this whole setup near a window. Okay, if you are going to perform this in the laboratory, you will put this setup near the window. Why window? Because the window gets the exposure of sunlight, and for the initiation of the process of photosynthesis, it is very much important that there should be the exposure of sunlight. Okay, and soon after some time, what you will observe is that the oxygen bubbles are coming out. These are the O2 bubbles that comes out. Got it? So therefore, as I have told you, that the liberation or evolving of any gas in the liquid or the fluid medium is very easier, because when a gas is liberated out into a liquid medium, bubbles form. So these bubbles are the O2 bubbles. When they form, basically when they are given out, we can infer that the photosynthesis is taking place within these hydrillas, and they are liberating the O2. That's all about the first experiment. Let's move on to the next one. So, this experiment or the demonstration deals with the product of photosynthesis. That is the first product. The previous one was dealing with the second product, that is the oxygen. This one is the main, the food, the production of food, that is the starch, is the end product of photosynthesis. I have already discussed with you all that we get C6H12O6 at the end of the photosynthesis, and which serves as the food for these plants. But is it so? So to check whether it is so or not, we perform this experiment, that whether the starch is the end product of photosynthesis. And for this, what do we need? We need a tripod stand, a lamp, okay, that is the Bunsen burner. This is the Bunsen burner, the tripod stand, a beaker, a test tube, and importantly, a leaf. Okay, so you may go to the garden and pluck out some leaves during the afternoon. Why we have selected the afternoon? Because during afternoon, the intensity of the sun is very much high, and the concentration of CO2 is also good. Actually, it's not about the concentration of CO2 or water, it's about the sun, that is the availability of the sunlight or the temperature. Okay, that is actually very much relevant during the afternoon, and at that time the rate of photosynthesis, that means the rapidity of photosynthesis increases in comparison to the other daytime. Okay, so that is why if the process or if the speed of photosynthesis when is increased, that means the product which is formed is also, it will be also so rapid, it will be also rapid, and it will be also increase the concentration of the product. Okay, so therefore we choose the afternoon to pluck the leaves out from the plant, and what we will do, we'll put the leaves into this test tube which contains alcohol. Now my dear students, can you tell me that why do we use alcohol? Why do we use alcohol? To decolor the leaf. For decoloration of the leaf, why do we use alcohol? Okay, leaf contain chlorophyll. What do you know? That leaves are green in color because they contain chlorophyll, and this chlorophyll is basically a protein complex and is soluble only in alcohol. Okay, that is why we use alcohol inside this test tube and we put the leaf into it, and then we will put this whole test tube along with the leaf and alcohol into a water bath. Water bath means we have taken a beaker half filled with the water. We'll put this, okay, and we will put it on the flame. Once you will put it on the flame and the water starts heating, the alcohol also starts heating. And due to the heating of alcohol, what will happen? This alcohol will rupture the chloroplast. What it will do? It will rupture the chloroplast. Okay, once the chloroplasts are ruptured, okay, when they are ruptured, the chlorophyll complex will come out and will dissolve into this alcohol. Simple. And the leaf that you will obtain after the boiling will be decolorized. Okay, so discolored leaf you will obtain. So we will take this discolored leaf into a petri plate. Okay, we will take this into a china dish or a petri plate, and what we will do, we will put few drops of iodine. Either you may put few drops of iodine, like 2 to 3 drops of iodine, or you may dip this whole discolored leaf into the iodine solution. Okay, you may put this whole discolored leaf into the iodine solution. Both things you can do. Either you can put a few drops of iodine onto it, or you may soak the whole leaf into the iodine solution. And at last, what inference you will find out? You will see that the leaf turns blue-black in color. And why so, my dear students? Why the leaf will turn blue-black in color? Because, do you know why? Because iodine, when reacts with the starch, okay, when you'll put iodine into the, any starch containing food, that turns it into this bluish black color. So this is the theory behind that. When the iodine is being put on a food which contains starch or on a starchy source, it turns the source into blue-black color, which confirms the presence of starch or glucose into it. Okay, Iodine turns the thing blue-black which contains starch. Okay. So that's all about these two experiments. Next, we are going to study about that the light is essential for photosynthesis and chlorophyll too. So this we will study in the next class. Thank you and have a good day.